Good morning, everyone, and welcome to the 2016 Royal Tyrrell Museum Speaker Series. Today, the Royal Tyrrell Museum and its cooperating society are very proud to present Dr. Kirstin Brink. Kirstin is currently a Killam and Michael Smith Foundation Health Research postdoctoral researcher in dentistry at the University of British Columbia. She grew up in a military family and living all across Canada and the UK. She obtained her bachelor's degree in biology and earth science at the University of Alberta and completed an honors project with Ruth Stockey looking at Cretaceous conifers from British Columbia. While completing her bachelor's degree, she worked 2 summers here at the Royal Tyrrell Museum, the first as a gallery interpreter and the second as a preparation technician. After completing her degree, Kirstin joined the staff here at the Royal Tyrrell Museum on a permanent basis as a preparation technician for 1 year before leaving to pursue her master's degree at the University of Calgary. Her master's research, under the supervision of Dr. Darla Zelenitsky, examined the pattern of growth and development of duck-billed dinosaur skulls from juvenile to adulthood. Following her master's, Kirstin moved east to pursue her PhD in the Department of Ecology and Evolutionary Biology at the University of Toronto, under the supervision of Drs. Robert Rice and David Evans. For her dissertation, Kirstin investigated the evolutionary change in the famous sail-backed Dimetrodon from the Permian period. Kirstin completed her PhD in June and has since moved to British Columbia to do a postdoctoral research on tooth development in living reptiles at the University of British Columbia. Over the years, Kirstin has done fieldwork in Alberta, British Columbia, Prince Edward Island, and Texas. Today, Kirstin will present the results of her PhD research on Dimetrodon, entitled Uncovering the Hidden Dental Diversity of the First Apex Predators to Identify Canada's Dimetrodon borealis. So without further delay, I present to you Dr. Kirstin Brink.
Thank you very much, Caleb. I'm really excited to be back at the museum and to get a chance to talk a little bit about some of the research I've been working on lately. So this talk takes us back about 300 million years to a time when everything looked really different. We have these really gigantic amphibians, it's really swampy, really warm, and probably the most intriguing thing is that there aren't any large terrestrial herbivores. There aren't any big animals that are eating plants. The only animals that are eating plants at this time are those giant insects. And if we follow this up about 10 million years, we start to see the first herbivores pop up in the fossil record. And these are animals that are actually able to eat plants and get a lot of energy, nutrition from those plants. But there's a snapshot in time in the early Permian where there are only a very small number of herbivores, and there are lots and lots and lots of carnivores. These are animals that are eating other animals. If we take a look at this about 290 million years ago, in yellow we have carnivores and omnivores, and just a couple of animals that are eating plants. And this is very different from any type of ecosystem you might think about today, where you have one top predator that feasts on several smaller herbivores or other smaller animals. So it's kind of perplexing to think, how might an ecosystem like this work? If we break down this chart into several smaller charts by formation, and we look at which carnivores are predominant, it is Dimetrodon. So it can form up to a quarter, up to about 60% of the animals that are top predators in these ecosystems. Dimetrodon belongs to a family called the Sphenacodontids. It is most commonly recognized by its really cool dorsal sail. And not only is it one of the most common fossils found in these formations, it's also the most speciose, so there's actually 13 species known. Dimetrodon has several cousins that are probably less well known. Some of them have the really elongate dorsal sails, some of them don't. Some of them are small, some of them have kind of intermediate height sails. And generally, overall, they have very similar skull shapes because these are all animals that are eating meat. They're all predators. And most of them actually lived at the same time in the early Permian. we'll take a look at where sphenacodontids kind of fit into the family tree. So within amniotes, these are animals that reproduce on land, there are two major splits. So in the yellow box is the diapsids, and the blue box are synapsids. In the yellow box, the diapsids include our reptiles, our dinosaurs, and our birds. And most people think that Dimetrodon might actually fall in this group, but it's actually along the stem of animals called non-mammalian synapsids. It's actually a really ancient ancestor of mammals. You can see it in the middle there. So if we take a look at these 13 species of Dimetrodon, 12 of them are actually known from Texas and Oklahoma and a couple of the other states in that area. And if we look at them in the stratigraphic column, so if we look at which species belong in which rock unit, we can see there's kind of an interesting trend of an increase in body size through time. There's a couple little hiccups to this problem. There are several taxa that co-occur. So these fossils are found in the same locality, in the same kind of area of rock. And when the paleontologists were first describing these animals in the late 1800s, early 1900s, if they found a small animal, they gave it its own name. If they found a big animal, they gave it its own name. And they never really thought that maybe smaller animals are babies, and middle-sized animals are teenagers, and the big ones are the adults. So nobody's really investigated this as to whether or not this is actually the true diversity we have of Dimetrodon, or if we have actually growth series. The other interesting point is that all these species of Dimetrodon are living alongside their other sphenacodontid cousins, like Tinospondylus and Secodontosaurus. So how are all of these top predators living in such large quantities in these kind of ecosystems that we don't really understand? And these are the questions that sort of drove my PhD research. What are the diversity of sphenacodontids in the early Permian, and how did they all coexist? And are there any significant evolutionary changes through time that we can see in the sphenacodontids in this little snapshot of Earth history where we're starting to introduce herbivores into the ecosystem? So what's happening to all the carnivores during this time? So I got to look at really, really, really cool fossils all across the United States. There were some really great Dimetrodon fossils. The big one at the top is at the Smithsonian. This guy at the bottom is at Harvard. There's stuff in New York and Chicago, University of Michigan. So really nice material to look at. And the first thing that I started to realize going through all this material is all their teeth look really different. So here's sort of a snapshot of all the different types of teeth that I saw within sphenacodontids. And I thought, this is really cool. Because teeth are often really well preserved. They're the hardest tissue in the body, so they fossilize really well. Teeth are common because these animals were continuously replacing their teeth throughout their lives. So they would shed a tooth, grow a new tooth, shed a tooth, and all those shed teeth are fossilized really well. Teeth actually provide really good characters for phylogenetic analyses, so to make those family trees. This is done frequently with dinosaur teeth. And the teeth also can give an indication of what the animal was eating. So different teeth are actually more effective at eating specific types of food. Another bonus is that some of the not so great fossils also had teeth. So these are a couple of the holotypes. So that means the names of these species of Dimetrodon were based on these fossils, which first time I saw them, I couldn't even tell what element they were. They're not that well preserved, but there are teeth hidden in there, which will help figure out all the relationships between these species. So we'll take a little kind of diversion into how a tooth is made. So in these reptiles, they're constantly replacing their teeth throughout their lives. But before that tooth is even formed, there's a little ball of cells inside the gums. And those cells will actually fold and they'll form the shape of the tooth before it starts to harden, before those tooth tissues harden. So in order to create a tooth that has lots of bumps on the side, those cells have to fold and form those bumps before the tooth will come out of the mouth. So to create a different shaped tooth requires a different developmental process. So we'll just keep that in mind as we look at some of these results. So the rest of the talk I'll split into kind of three sort of stories. The first one will look at the tooth crowns. That's the part of the mouth or part of the tooth you can see when you open your mouth. I'll talk a little bit about tooth roots. That's the part inside the gums. And I'll talk about how these tooth characters actually helped to figure out a little Canadian fossil mystery. So we'll start with the tooth crowns. This is work that I published a few years ago, featuring paleo art from the Royal Ontario Museum's paleo artist, Daniel Dufault. So the name Dimetrodon means 2 measures of teeth. And this is because if you think of a reptile like any small kind of lizard, all their teeth sort of look the same within the mouth. But Dimetrodon actually has varying sizes of teeth, and that's what the name refers to. So there's these really large teeth at the front of the mouth, and then a big gap. And that's actually a gap to accommodate the teeth from the bottom jaw. When they close their mouth, they'd have really strong grip on whatever they were trying to eat. Sometimes there's a little funny little tooth at the back of that gap, and then these really large, what we called caniniform teeth, that also help to improve that grip. And a series of smaller teeth along the back of the maxilla. And some of these teeth have these little bumps along the fronts and back of the teeth, kind of like a steak knife, and they're called denticles. And this is really cool because these denticles can be different shapes and different sizes in different animals, especially in dinosaurs. So a lot of people who work on meat-eating dinosaurs love to look at tooth characters and figure out what's the relationships between those animals or what were they eating. So I was excited to know this and to think, oh, maybe this might be good characters to use to figure out what's going on with Dimetrodon. So these are the questions I wanted to address with this part of the project. So are there differences in the tooth structure between species, and might they be useful to help figure out which ones are which? And are there any patterns that change through time within this group? So these are the samples I got to look at. So 3 different kind of time points in the Dimetrodon series, and then some of those co-occurring taxa, Tenospondylus, Secodontosaurus, and Sphenacodon. So we'll start with the youngest and largest sample, because these teeth are the ones you saw. These are the Xiphonont teeth. So they have a kind of blade-like shape in lateral view. There's little dents called a sulcus on the tooth. And then you can see the denticles along the side. And these are actually, there's been lots and lots of studies done on how efficient a tooth like this is at biting meat. And they're actually really good at ripping through prey and slicing off chunks of meat. So this is a good indicator of a really efficient tooth, good at eating meat. And if we cut that open to sort of look at what the inside of the tooth looks like, so this is a slice through those denticles, we can see there's kind of a wiggly pattern in between the 2 sort of rainbow colors. So if we remember the way that a tooth develops, those cells, before the tooth even forms, have to fold into that kind of ribbon pattern in order to make these little denticles. Okay, we'll move up through we'll move down in section to look at the intermediate time point species of Dimetrodon. And these ones, they have little bumps along the side too. And some of them actually can curve around the back of the tooth to look sort of like little cusps that we might see in other mammals. But when you if you look into the edge of the tooth, the bumps look a little bit smaller than the teeth we just looked at. And when you cut them open, you see you don't see that wiggly pattern anymore in between the junction where those 2 tissues meet. It's a straight line between the dentine and the enamel. So what we're seeing here is a different developmental process. So the shape of that tooth isn't being formed before it's erupting. It's actually being formed by how much enamel is deposited on the outside of the tooth. So different developmental process to get a similar looking tooth from the outside. And if we go to our oldest species, we don't see any of these bumps at all. There's these tiny little cusps at the very bottom of the tooth, kind of like the intermediate species. But the edge of the tooth doesn't have any of those denticles or any enamel serrations along the edge either. And if we look at this pattern overall, what we're seeing is almost like a progression from simple tooth to kind of intermediate tooth to a more complex tooth through time, just in Dimetrodon. Now, if we take a look at the animals that are also living at the same time, these might be a little harder to see, but Sphenacodon on the left, the teeth overall look pretty similar to Dimetrodon, but the carinae, the edges of the teeth, are very smooth. And in Tenospondylus, it's the same thing. The edges of the teeth are very, very smooth, but there are a couple little ridges that kind of circle the tooth sometimes. This is Secodontosaurus, and it has what I'm calling wavy enamel. So if you look at the image on the bottom right, that junction between the dentine and the enamel is straight. There's no folding in the shape of the tooth. So all of those wrinkles are created by just enamel only, different deposition of enamel crystallites. Now, if we plot all of these patterns together back into the stratigraphic column, we see where these species overlap, they all have a different tooth type. So what that means is that these teeth are probably, they're more efficient at eating particular types of prey if you have a specific tooth type. And this means that maybe each of these animals is eating something different, which actually helps them coexist in the same environment. If we dive into more detail into just what this means for Dimetrodon, perhaps maybe the smoother teeth, the smaller animals are able to eat any of the amphibians that are around. You add a little bit of complexity to the tooth, maybe they're actually able to eat some of the larger amphibians that are around at the time. And the coolest thing, when we see the appearance of those true denticles, is also when we see in the fossil record the first appearance of the really large-bodied herbivores. So maybe this means that we have a more efficient tooth and they're actually able to eat any prey animal that they find at this time point, which is pretty cool. So to sum up this part, there's a large diversity of tooth morphology in sphenacodontids, which reflect differences in prey selection. And tooth shape changes through time in Dimetrodon, but not those co-occurring species. And the change in tooth shape actually allows for more efficient predation in later species and more efficient food handling. Okay, we'll jump down the tooth or up the tooth, depending where you are in the mouth, and take a look at the tooth roots. And this is a project that I worked on with my lab mate in Toronto, Aaron LeBlanc. So when Cope was first describing these specimens, he gave pretty detailed descriptions. And in one paper, he said, there's this tooth, and the base of the tooth looks like a figure 8 shape. And he kind of left it at that. And in case another paleontologist re-examined these specimens some years later and said, nah, it's not a figure 8, it's just a crushed tooth. So I put an arrow at that point on the tooth of the specimen that they were both describing. And kind of thinking, I don't know if that's crushed or I don't know if that's a figure 8 or what, but I kind of figured it might be worth looking into. Since the tooth crowns have these interesting characters, there might be more going on here. So at the same time, my lab mate Aaron was working on this concept of plisodentine. And all that really means is folded teeth or folded dentine. So in several types of fish and amphibians, if you cut the tooth open, and look at that picture with all the black squiggles, that's all dentine. And it's basically just folded up dentine inside the tooth that helps to strengthen it. And it was originally thought that this character was only found in fish and amphibians, but a lot of recent studies have discovered them in several extinct groups of amniotes, and some reptiles have it, Komodo dragons have this folded teeth, but it's never been reported in synapsids before. So these are the questions we wanted to address. Is plisodentine present in Synapsida? Is it a figure 8 or is it crushed tooth? And if it is present, do we see any other kind of signals? Is there a functional signal or a taxonomic signal in the shape of these tooth roots? So once again, the sample that I looked at 2 different time points of Dimetrodon, and then the co-occurring taxa Cegodontosaurus and Sphenacodon. And I'm going to present these results in the form of this family tree. So we'll start at the bottom with Ianthodon. This is a really small sphenacodontid. So what I'm showing here are sections cut through the tooth root in this direction, transverse direction. And you can see there's these really cute little pinched folds at the front and the back of the tooth. And it gets progressively kind of wigglier towards the base of the tooth. And we're like, oh, this is good. This looks like folded dentine. So I'm going to plot it on this family tree showing the shape of the tooth root, and then the colors correspond to those enamel characters from the tooth crown project. This is Cegodontosaurus. And it kind of, sort of, maybe has a couple of folds from the top and bottom of the tooth. Kind of, not really, but it's not as pronounced as it was in Ianthodon. So I'll plot that on the tree. Next, we'll look at Sphenacodon. And this is where it kind of gets cool. This tooth is crushed, unfortunately. But when you reconstruct it, it forms a really cute 4-leaf clover shape that we were calling it. So put that on our tree. Next, we'll have a look at Dimetrodon. So the older species of Dimetrodon has, once again, that 4-leaf clover shape. And we found it in pretty much every tooth position along the top and the bottom of the jaw. So we knew that it wasn't just maybe some teeth in the mouth had it, or maybe only some specimens had it. We looked at several different animals, and they all had this really cool four-leaf clover shape. And then when we looked at our largest and youngest species, we didn't see the folds. So we looked at several different specimens, we looked at different depths in the tooth root, and there weren't any folds in that denting. And we thought, this is interesting. So what's going on here? So if you notice, if you look at the little cartoons on the right, all of the teeth that have the folds in them, the roots are very short. And if you look at the bottom species of Dimetrodon, it doesn't have any folds, and the tooth root is really, really long. So what this could mean is that the teeth are under such incredible stress when the animal is biting into something. The tooth has to be really well anchored into the jaw. So if you have a really long tooth, but a really short tooth root, by adding those folds into the base of the tooth, you're actually increasing the surface area for that tooth to be stuck into the jaw. So if the later occurring animals, they're much larger. Their skulls are actually a bit taller, and they have much more room for a really long tooth root. And they don't need to have those folds in their tooth because they're getting all their attachment space by height instead of by folding. So that's what we think is happening in this group. If you look at the pattern within Dimetrodon again, the older species have the four-leaf clover-shaped root, and they have the enamel serrations only on their teeth, where the larger, younger species have the rounded, really long tooth root, and they have those true denticles. So we're thinking this is a more efficient tooth in the top of the strat column. So let's sum up this little part. This is the first documentation of Plisodentine in Synapsida. It's the folded tooth roots. And it's the first time that the loss of Plisodentine has been documented within one taxon, or in this case, it's Dimetrodon. The folds in the root appear to be, the different shapes of the folds actually appear to be a good way to identify different species from each other. And the Plisodentine helps to strengthen teeth in those animals that have the really short tooth roots. Okay, so now we're armed with all this information on tooth crown morphology, tooth root morphology, so we can try to apply it to kind of a little case study. And this is a fossil. It's called Bathionathus borealis from Prince Edward Island. This is an image that was drawn of it when it was originally described in 1854. And it has a really cool history that I'll talk a bit about. But I wanted to kind of re-examine this fossil now that I knew the teeth could be really useful for figuring out relationships among the sphenacodontids. So in the early Permian, this is a map of the paleo-equator at the time. So that line running through the map would have been the equator. And those black blobs where the black arrow is are where fossils of sphenacodontids have been found. And the red arrow at the top is Prince Edward Island. So it's just south of the equator. And all the Dimetrodon from Texas are also just south of the equator. These are some of the fossils that have been found from Prince Edward Island over a really long time. The fossil record is pretty spotty on the island. Most of the material is found in really big boulders or small little chunks of rock that erode out of the cliffs along the oceans. So a lot of it's really broken up and kind of hard to find unless you're there at the right time. So like Langston actually spent, I think, 4 or 5 months on the island looking for fossils and was able to find 10 things that maybe could be described, where if you go to Dinosaur Park in Alberta, you could find hundreds and hundreds of fossils in one day. So there's not that many fossils, but when you do find them, they're pretty good. There's lots of footprints, like on the bottom left here. The most recently described was almost a complete parareptile at the top right, called Erpatonyx. It's probably the most complete fossil ever found on the island. And there's also lots and lots of really good plant material. But my favorite is Bathionathus. This one was actually found more inland. It wasn't found along the coast, as most of the rest of the material was. So it was found by a farmer who was digging a well on his property, looking for water. And this specimen, once it was found, has probably the most interesting history of any fossil I've ever looked at in terms of players in the game. So it was found by a farmer, Don McLeod, 1845. And at the time, he knew there had been a geologist on the island looking at all the fossil plants by the name of Dawson. He worked at McGill in Montreal. And he approached Dawson and said, hey, I found this thing. I want to sell it. I don't want it. And Dawson was like, I like plants. I don't really care about this fossil, but I'll try to sell it for you. I'll try to help you out. And it just happened at the time that Sir Lyell, who was probably the most preeminent geologist at the time from the UK, was visiting Nova Scotia and Prince Edward Island looking at the fossils. And he advised Dawson not to send it over to the UK, where most of paleontologists were working at the time, but to try and keep it in North America. So Dawson had wanted to sell it to this guy Owen, who maybe you heard he actually named the term Dinosauria. And so he knew his fossils, so he would have been probably the obvious choice to send it to. But there's actually some correspondence that was found later that Lyell and Owen had been fighting at the time. So Lyell discouraged Dawson from sending it to Owen because he was mad at him. So anyways, the fossil ended up going to the National Academy of Sciences in Philadelphia, where Leidy was working. And Leidy was a super cool guy. He was an anatomist. He worked a bit on paleo. He was a criminologist. He solved the first murder, or helped solve a murder crime by identifying the differences between chicken blood and human blood on somebody's stained apron. Yeah, he was a cool guy. So they sold the fossil to Leidy, I think for, it was 10 pounds, I think, because at the time Canada hadn't yet switched over to the dollar system, which is worth about $3,000 today, I think. So Leidy got the fossil. He described it to Bathynathus borealis in 1854. And he described it that because he thought it was the lower jaw of some kind of large bipedal reptile. He never actually said dinosaur, but compared to the fossils that were coming out of the UK at the time, probably a dinosaur. So after that, paleontologists working in North America, describing lots of dinosaur fossils, probably some of the most famous researchers who worked during the dinosaur rush, Marsh and Cope, both refer to Bathynathus in different publications as a dinosaur. And then it wasn't until 1876 when Owen gets back in the game, and he had been describing several fossils from South Africa called Theriodonts, and these are part of that lineage in the synapsid line. And he was like, hey guys, you know what? If you flip this fossil over, it looks like the upper jaw of these synapsids that I'm working on in Africa. And everybody was like, I don't know, it's probably a dinosaur. A couple more years later, in two separate publications by Von Huene and Case, European and American researchers, they said, yeah, Owen is right, this is probably a sphenacodontid maxilla. So they flipped it over, and it's been considered a sphenacodontid maxilla ever since. So several researchers have worked on it in the 1900s. Romer, Langston, he was the one who went to PEI to look for more fossils. Robert Rice, my supervisor in Toronto, worked on the specimen. And they all said, yeah, it's a sphenacodontid, but we don't really know where it fits into this tree. We don't know if it had a sail like Dimetrodon. We don't know if it was one of the more closely related to some of the maybe more boring cousins that don't have a sail. Here's a little bit of trivia. Bathynathus was the second vertebrate fossil named from Canada. So can anybody think of what the first one might have been? I'll just show you. It's Dendropodon. So this is a small amphibian that Owen described when Lyell brought it back to the UK during one of his visits to Nova Scotia. And it was named 2 months before Bathynathus, which is kind of funny, in the same year. What I think is interesting is it was one of the first specimens to have a histological examination done. So that means they cut the bones open and looked at the sizes of the cells inside the bone. And amphibians have a very distinct cell size and shape. So they're able to actually confirm that it was an amphibian by looking inside the bone. This was in 1854. That's pretty cool. So some trivia to go tell your family and friends. So even though we didn't really know exactly where Bathynathus fit into the sphenacodontid tree, it's been reconstructed looking like Dimetrodon for a really long time. This is a coin the Canada Mint put out a couple years ago. I think Francois actually helped write it up and get all the facts straight for this one. So, and it just looks really cool, I think, reconstructed as Dimetrodon, which is why people would often do it that way. So the point of this part of the study was to see, looking at all these tooth characters that I now have, can we actually figure out where it fits into the family tree? So this is what the fossil looks like when it's flipped the right way. It's part of the upper jaw with several teeth in it, and that's what it looks like kind of overlain onto a Dimetrodon reconstruction. If we take a look at the teeth, it has that same sulcus as Dimetrodon has, and there's no evidence of a fold along the base of the tooth. If we take a look at the denticles on the teeth, I wasn't allowed to section this to cut the specimen open, but that's okay because the enamel was a little bit clear, so we were able to see there's actually that kind of ribbon folding around each denticle. So these are true denticles, it's good. The specimen is fortunately or unfortunately broken in half. So we're able to see the length of the tooth root looking kind of into the maxilla. And the root is longer than the crown of that tooth. We wanted to just double check this, so we put it through a CT scanner in Toronto and to look at the shape of the tooth roots. And highlighted in red, you can see they're round. There was no evidence of any of that four-leaf clover-shaped fold in any of the scans. Here's a reconstruction of what the teeth looked like in the CT scan. And the little parentheses show that the length of the root is longer or equal to the length of the crown in most of the teeth. So if we plot these, if Bathinathus into the family tree, considering these tooth characters, not really surprisingly, we find that it looks very, very similar to that species of Dimetrodon that also had the rounded tooth roots with the true denticles on it. And it's kind of tucked up right nicely into the Dimetrodon family. So usually when this happens in these family trees, it's usually the simplest thing to do is just to change the name of that one species so that the whole family has the same name. So I wanted to change the name to Dimetrodon borealis. But this introduces a whole new problem in terms of the way that fossils and animals and plants are named. The name that is given first has priority. So if a name that is named later wants to be collapsed in with one of those previous names, it's the first name that has priority. So in this case, because I'm syncing those two together, everything should be called Bathinathus, and there should not be any Dimetrodon. So it would be kind of like a whole Brontosaurus or Pluto situation again, where Dimetrodon didn't exist anymore and nobody would talk to me ever again. So in order to fix this problem, I had to write a report to the International Commission of Zoological Nomenclature. And these are the people who control the rules over how animals are named. So this is a petition that's online right now. So the commissioners consider it. They post it online, and then people, all of you guys and researchers, anybody, can post online and say whether or not they support this suggestion to make Dimetrodon the name with priority. And this is posted, I think, for about a year. And after a year, the commission looks at all those comments. Some of them get published in their journal, and then they make a decision as to whether or not Dimetrodon would have priority or not. So technically, right now, Dimetrodon doesn't exist. Everything should be called Bathinathus right now, unless this gets approved. So fingers crossed. So to kind of sum up everything I've talked about today, I found that tooth characters are pretty useful for distinguishing between species of sphenacodontids. And different sphenacodontids coexisted by eating different prey, as interpreted through tooth crown and tooth root morphology. And overall, the improvement in carnivorous adaptations throughout the early Permian means a more efficient and more effective apex predators. That's our Dimetrodon eating a large amphibian. I have very, 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 very many people to thank for this research, and thank you all for listening.